going to speak about basic science. I'm the only one who's going to do that today. Everyone else is going to talk about surgery, interventions. Oh, the rats? Okay. So, we as geneticists, and I'm a cardiologist in training, but in passion I'm a geneticist, are interested in looking at the pathophysiology of diseases by looking into, we take our animal model are humans. So we look into human families with some disease trait that we like to look at, and then we try to describe the phenotype, and by describing the phenotype, we can go ahead and find the gene. The genes are helping us to develop animal models which help us understand the pathophysiology. And unlike any other animal models, we know the animal model is relevant because it's depicting the human genetic error that's making the disease. And our end of the circle is when we try to develop therapeutics that help prevent or treat the disease. And I'm going to show you two examples of that today by looking at the genetics of mitral valve prolapse and aortic stenosis. So the first story is mitral valve prolapse. I don't think I need to talk much about what's the disease in this audience. This is a very common disease, about 2.4% of the population. It's associated with many side effects, including mitral regurgitation, congestive heart failure, thromboembolic phenomena, and even today a lot of publications are coming out connecting this disease with sudden death. And so when you look at the MVP, we found this family many years ago. This is a large pedigree segregating MVP, meaning that there are a lot of individuals within this family that have MVP. It looks like an autosomal dominant disease because it's going from one generation to the other. And by using linkage analysis techniques, we were able to localize the mutation into a small chunk of a chromosome 11, which we call a loci, and we knew that the disease is segregating with this loci within the genome. Today, there are advances in the ability to perform DNA sequencing. You can do a lot of sequencing with technology called massive parallel sequencing or next generation sequencing. So we use this technology to download the region where we thought the mutation was in 4 individuals from this family, and to sequence the entire region in search for a mutation. So when you do such massive sequencing, you end up with a lot of potential mutations. So we ended up as enigma. Okay, so we ended up with 5,000 potential mutations, but only 2,000 were shared by all 4 individuals with MVP. Of these 2,000 mutations, only 155 were not described before in any previous database, and of these, only 6 were coding sequences, meaning that they're changing the sequence of the protein they are coding. Of these 6, only 3 were able to validate using regular sequencing technology. And this was in 2 genes. One is called DCHS1 and the other is called APBB1. And to prove that these are indeed mutations, we use a zebrafish model. We use animal model. Today, a lot of people are moving from mice, which are very difficult to handle and to generate, to zebrafish. What's neat about zebrafish is that they are transparent. They're very similar to the zebrafish we have in our homes in the aquarium. I don't have an aquarium. I hate this, but because all fish die. But anyway, in animal labs, you can take the fish, they're transparent, you can put them under a microscope and look at the foaming heart and the way it's functioning. And also another neat thing about this is that you can knock down any gene within their genome very easily. So here is a normal foaming heart, and you can see the blood is flowing in one direction from what's going to become an atrium. The heart is looping, and then the ventricle, and then it's going into the body. But when you knock down DCHS1, one of the genes we found, you end up with a very severe phenotype where the valve is not forming, and you can see actually how the heart chambers are dilated, and you can see the blood going back and forth between the heart chamber, meaning that when knocking down DCHS1, there is a severe phenotype. And when you transfect these zebrafish with this severe phenotype with the regular human RNA, you get reversal of the phenotype, so there is no phenotype. But when you transfect the fish with the mutated human RNA, you get nothing. This is a proof that the mutation we found is pathogenic, because it's not salvaging the phenotype we generated in zebrafish. And it didn't happen for the other gene, which is APBB1. And then when you look at knockout mice for DCHS1, you find a phenotype, or you find pathology that is very much like myxomatous degeneration. So here is a normal valve on a normal mouse. This is the posterior leaflet, anterior leaflet. This is the aortic valve, and here is the chamber muscle. And you can see that the valve is thin, it's not elongated, and you can see the layers of the cells are very much arranged in the right way. When you knock down one allele of DCHS1, you end up with something looking like very much myxomatous degeneration, because the leaflets are elongated, thickened, and there is a mixture of cells within the matrix of the valve, which is what we call myxomatous degeneration. And similarly, we can see the MVP in echo of the mice. So there is an echo from a normal mouse. You can see this is the atrium, this is the ventricle, this is the valve. And on the knockout mouse, you can see that the posterior leaflet is prolapsing. So the phenotype you get on mice knocked down with DCHS1 is very similar to the human disease. So what is DCHS1? DCHS1 is an important protein for epithelial surfaces. Cells in epithelial surfaces are aligned together in a certain way. The best analogy for that is hair. I'm not a good example, but anyone with hair knows that hair grows within a certain direction. It's not just random. All hairs go in one direction. How does the hair know where to go and where to grow? It knows because cells are connecting with each other. They are handshaking. And these genes that are handshaking each other are important for the normal matrix of epithelial cells. So when you disrupt that, you get an abnormal epithelial layering. And indeed, when you look at the cells of knockout of DCHS1, so here is a normal mouse, H&E staining. Here is a vector map showing the alignment of cell, and here is a zoom on this vector map. So you can see that the more you disrupt DCHS1, this is the heterozygote form mutation, this is homozygote, the cells are more and more misaligned. They don't know where they should be within the matrix, proving the concept that the mutation causing myxomatous degeneration is something interfering with cell-to-cell adhesion. So we were very fortunate to publish this in Nature this year. And then we went on and published and did another study. This I'm using a different genetic approach called genome-wide association study, GWAS. So what's a GWAS? The GWAS is in many ways an epidemiological study. You look at a cohort of patients, very large group of patients, and you compare them to control. But unlike regular epidemiological study, when you most of the time look at the environmental exposure, for example, sun exposure in melanoma or smoking in lung cancer, you look at changes within the DNA, and you hope to find changes that are significantly more prevalent among the patients. And today with the technology, you can genotype almost 5 million different SNPs. SNPs are single nucleotide polymorphisms. And what you hope to find is something that is way more prevalent within patients to assume that it's causing the disease. This is generating a statistical problem because this is an excellent example of multiple comparison situation. So to know that you didn't find anything by chance, you need to replicate your finding in a second cohort, and then ideally to find the animal model that proves that you found a mutation. So our discovery cohort was from France, unfortunately, because we were battling between us and the lab. I worked in the USA. Who's going to get the first 1,000 patients, and whoever got first was the one who was leading the study, and so we lost this fight, and therefore the French are the first authors on this publication. So the discovery cohort was from France, the replication cohort was mostly from the US, and we found two genes that are associated with MVP. One is called tensin-1, and one is called LMCD1, and we were able to prove that in zebrafish again, so we found two more mutations causing MVP. And we were, again, fortunate to publish this in Nature Genetics this year. So what is the common denominator of all this? On normal cell surface, or normal valve, there are two epithelial surfaces. One is called the atrialis, and one is called ventricularis. And in between them, there is matrix composed mostly of mesenchymal cells, which are alpha smooth muscle cells or fibroblasts, that secrete matrix that should compact, align well, and form the normal valve. There is a process that constantly is going throughout life during valve development and through life called EMT, endothelial mesenchymal transformation, which is important for valve maintenance. In this process, epithelial cells go transformation and change from epithelial cells into mesenchymal cells, migrate into the interstitium, secrete matrix, and this matrix should compact and maintain the normal structure of the valve. Whenever you disrupt any of this mechanism of either cell transformation or migration, you may end up having a valve phenotype. And indeed, when you look at the proteins that have been linked to this important process of EMT, some of them have already been associated with valve pathology. For example, Notch1 is the first gene that has been related to bicuspid aortic valve, and TGF-beta signaling is very important for Marfan disease and mitral valve prolapse. And maybe we'll have news next year on these genes from our lab in Jerusalem. So in summary, we found 3 genes, one using family linkage analysis and the other genome-wide association study causing MVP. MVP is a genetic disorder, and I tend to think that it's I know that it's been caused by many different mutations in different genes. It's not one gene, one disease. And we think that the mechanism of MVP is disruption of normal valve homeostasis, where epithelial cell migration into the mesenchyma EMT process is disrupted. But I think the most important message for a clinician is that the valve is constantly changing and reforming and deforming and responding to various external stresses. And if we can understand these mechanisms, we can perhaps develop treatments for diseases causing valve pathology. The second story I want to tell in short is the story of the aortic valve stenosis genetics. So 3 years ago, the first genome-wide association study on calcific aortic valve was published in the New England, and a very surprising gene came up as the one causing or associated with aortic stenosis. And this gene is lipoprotein lithium A, LPA, which we all know from atherosclerosis. This is a gene that high levels of it may cause atherosclerosis. So what is LPA? LPA is this protein which has lots of Kringle repeats. These are the Kringle repeats, I think, attached to LDL particles, and we know that high levels of that are causing atherosclerosis. Its role is not really known, but we do know that it's interfering. It's very similar to plasminogen, and therefore it's inhibiting plasminogen activation, and therefore it's also linking the atherosclerosis process to thromboembolic events. So the question is, can we use this knowledge to change the course of the disease? So first of all, can we reduce the levels of LPA in the... I'm sorry, one more thing I wanted to say, that before the genetic study, a lot of people have speculated and studied many different mechanisms causing aortic stenosis. And so this is just one review of that out of many. No one has suggested or thought that LPA is associated with the disease. This was something that needed the human model to find and to go on. But since this publication, there is a question whether reducing LPA will improve the outcomes in patients with calcific aortic valve stenosis, and we don't know the answer yet, but we do know that we do have tools to reduce LPA. One of them is PCSK9 inhibitors. So PCSK9 inhibitors, which we all are going to use very soon, very often in patients with high cholesterol, are very strong LPA reducers, so they can reduce the levels of LPA in 30%. But the story of LPA is not just that, because LPA has a role in scavenging oxidized phospholipids in the plasma by an enzyme associated with it, lipoprotein-associated phospholipase A2. If you can recall, this is one of those markers of inflammation related to atherosclerosis, like high sensitivity CRP. It's very similar. There is the plaque test. It's the same thing. So this protein is linked to LPA. And if you look at patients with aortic stenosis, you can see that the progression of the disease is very much related to the activity of phospholipase A2 in the plasma. So if you have low activity of phospholipase A2, the progression of aortic stenosis is lower than patients with high LPA activity, a high PLA activity. So there are ways to reduce PLA activity in the plasma. There is a drug called daraplatinib. I have to learn these. I promised you a practice last night, but apparently not enough. Anyway, so there is an agent that reduces the activity of PLA2 in the plasma, and there is also a new company that has developed antisense that reduces the level of LPA into almost zero. And I think they're planning to use it in aortic stenosis as a medical treatment for the disease, preventing perhaps the need for more aggressive interventions. So just to summarize that, we learned from genome-wide association study that that lipoprotein LA is a risk factor for aortic stenosis, and there are ways to reduce the levels and the activity of LP-LA, and so I think within the next near future we will know if these are affecting the progression of the disease. But it makes a lot of sense to try these drugs. So some of them are available in the market already. This has been a journey by the International Mitral Valve Do Consortium, which when they came here 4 years ago, we started this tradition of valve meetings in Israel, in Jerusalem. And I want to thank my lab in Jerusalem, particularly two people, Idit Gaba, I hope they're here, and Amichai Goodgold, our students that are working on these projects. And also to say that we have funding. Professor Lotan is sitting here, he'll provide the funding. הנה, הבטחתי, עכשיו אין ברירה. And we're hoping for collaboration. We want to do science. It is interesting. We have the advantage in Israel of having a small country with large families. And if anyone wants to come working with us, or that we will help train students, and you have families segregating any valve disease, we're happy to collaborate, to help, to support. We have funding, and we can help with that. And this is the Ledoux Consortium when we met in Boston many years ago. I'm here at the end. But she's beautiful. I stood next to her. So. Thank you.